Hello, good morning. Can you hear me?
Yes, sir.
Okay. Can you see the slides screen?
Yes,
Yes,
sir.
sir.
Okay, right. So good morning, everyone. So this lecture actually I have been doing during last 10 years. Uh, this is our topic is carriage of oxygen and carriage of carbon dioxide, control of ventilation, and hypo and hypercarbia and fetal oxygenation. Right. Then how can I remove this one? Uh, right. Uh, Now, carriage of oxygen. So now we need average adult, that is 70 kilogram person need 250 ml of oxygen per minute at rest. That is at rest, but in during exercise and various other situations, you need even very high oxygen uh, amount is required. But this is at rest. The oxygen is carried in the blood in two different forms. So that is the dissolved in the plasma, so which is responsible for the partial pressure of oxygen in the blood. So it is 0.003 ml per millimeters of mercury per 100 ml of blood. The actual volume is that for 100 ml in 100 ml of blood, uh, very minute amount. So with this, you can't supply this 250 ml per minute, right? So the other form is the bound to hemoglobin. The oxygen bound to hemoglobin is the other form, which is more important. So for the carriage of oxygen. Because now let's see how it is, why it is so important to have hemoglobin for the oxygen carriage. Now, I said the dissolved form is only 0.003 ml per 100 ml per millimeters of mercury. Uh, so, uh, so only now you can see from this calculation. So very minute amount, right? So zero point 0.3 per 100 ml. So into 100 millimeters of mercury. So in the partial pressure, uh, right? So in the blood is 100 millimeters of mercury. So therefore, so 100 into 100 ml. So then it becomes 0.3 ml per 100 ml when the partial pressure is 100. So that is in 100 ml. So you have to convert it to 5,000 ml, which is the usual cardiac output at rest in an adult, average adult. So then in zero point 0.3 ml in 100 ml. So then 1,000 ml it is 3 ml. So 5,000 ml. So it will be 3 into 5. So that is 15 ml. That is the total amount that can be carried in the blood when the partial pressure of oxygen is 100 millimeters of mercury, and then when the cardiac output is 5 liters. So there is the amount that can be carried in dissolved form. So, but you need 250 ml for 1 minute. So therefore, now, right, so this is what I said. So, yeah, 250 ml is the demand, the supply is 15 ml from dissolved oxygen. So that is grossly inadequate. So if you look at the oxygen flux, so what is the definition of oxygen flux? The amount of oxygen leaving the left ventricle. So amount of, yeah, left ventricle per minute in the artery. But there are various other definitions, similar definitions are there, the amount of oxygen supplies to tissues in 1 minute away. But the same thing is defined in various different definitions. So now that is the other definition. You can see amount of oxygen delivered to tissues per minute. So whatever the definition, the same, they refer to the same concept. So what are the factors affecting oxygen flux? These are the factors affecting oxygen flux. One is cardiac output, the other one partial pressure or the saturation, and the hemoglobin percentage. So these are the factors affecting the oxygen flux. Right. So let's calculate the oxygen flux. Right. I said cardiac output in average adult at rest is 5,000. Cardiac output into percentage saturation into hemoglobin percentage into 1.34. Now you have to add this the dissolved oxygen fraction also to this equation to complete this oxygen flux. So that is 15 ml should be added, right? So not only bound to hemoglobin, so it is carried in the plasma also, as I mentioned before. So therefore, actually, you have to add that 15 ml also to this one. So this is the amount of oxygen carried in the hemoglobin, right? So the cardiac output is 5,000 ml because others are in milliliters. So we have to convert the cardiac output into milliliters. So cardiac output is 5,000 milliliters. And the saturation, so almost 100%. So for convenience of calculation, right? So I have made it, usually 97 to 100. So I have taken as 100, right? 100 out of 100. Then when you cross, so you are left with one there, right? Then hemoglobin percentage is 100 milliliters. You have 15 grams of hemoglobin into 1.34. So what is this 1.34? Anybody? Right? What is this 1.34? Anyone?
1 gram of hemoglobin can bind 1.34 oxygen.
Amount of hemoglobin, how much hemoglobin?
1 gram of hemoglobin.
Yeah, 1 gram. Yeah, you are correct, right? So it is the amount of oxygen carried that can be carried by 1 gram, right? 1 gram of hemoglobin, right? So that is the amount. But actually now it is not 1.34, right? But it is 1.39, right? Actually, strictly speaking, it is 1.39 can be carried in the hemoglobin. So 1 gram of hemoglobin can carry 1.39. But there's a difference of 0.005 because there are hemoglobin molecules that cannot bind with the oxygen. What are those hemoglobin? Can you name some? Like this carboxyhemoglobin, right? Carboxyhemoglobin, right? Uh, various impurities are there. So because of that, actually only 1.34 can be carried by 1 gram of hemoglobin. So hemoglobin percentage is 15 gram in adult male average. So taken as 15 ml. Then when you multiply this it will come to 20 ml in 100 ml of blood. 20 when the hemoglobin percentage is 15 gram and the saturation is almost 100%, so it will come to 20 ml of oxygen, right? So in 100 milliliters. So then it finally it comes to 1,000 milliliters. So 1 liter of oxygen in the blood, right? So this is the amount of oxygen available to the body in 1 minute, right? When the cardiac output is 5 liters and the saturation is almost 100% and the hemoglobin is 15 gram percent, so you have this amount, right? Plus the dissolved oxygen, that is 15 milliliters. So, right, this is when you compare with this 1,000 milliliters, so that 15 milliliters is a negligible amount, right? Negligible amount, right? Uh, Now, this is the oxygen dissociation curve. All of you are familiar with this oxygen dissociation curve, right? So you have you should now this is a frequently asked question in the Viva. So you should be able to draw the oxygen dissociation curve and then discuss about the oxygen dissociation curve. So in the x-axis, so the partial pressure of oxygen, now here it is in kilopascal, and but you can take millimeters of mercury also there. So on your y-axis, the percentage saturation of hemoglobin with oxygen. Right. You can see here that when the partial pressure of is 100, so about 13.5 kilopascal, so the saturation is almost 100%. So that is in the arterial blood. Arterial blood, you have about 100 millimeters of mercury partial pressure with almost 9900 saturation. So that is called the arterial point. Then in the venous point, so you have 75%. That is actually mixed venous blood. So you have 75% saturation. So the partial pressure is about 40 millimeters of mercury in the venous blood. Partial pressure 40 millimeters. But if you take different organs, so you will have different venous saturation. So now if you take the kidney, renal vein will have a higher saturation because the kidney is getting about 20% of the cardiac output. That is not for the metabolic demand, but it is for the excretory function. So it needs such a big amount of blood. So therefore, venous saturation is high in the renal vein, whereas in the hepatic vein, so it is comparatively even lower than 75% because it gets portal blood also mostly. Right. Therefore, the venous saturation in the hepatic vein is very low. So like that, so you can have differences. But finally, when blood comes to the mm, when blood comes to the pulmonary artery, so it will be nicely mixed with other areas. So roughly it will have 75% saturation. And then the other important point in the oxygen dissociation curve is the P50. So you should be able to define P50. So it is now you draw a line from here, 50% saturation, until that line cross the curve. So from there you draw a vertical line until it meet the x-axis. So that is the P50. That value is called P50. Normally it is 26.7, somewhere around that region. So what is the purpose of talking about this P50? Why are we talking about P50? Why are we talking about P50? What is the importance of P50? Anyone?
One thing is it gives us a reference point to draw the oxygen dissociation cursor, and then it also gives us an idea of the affinity of that particular hemoglobin to oxygen, whether the oxygen will dissociate or not uh, at a particular level.
Yes, I think yeah, you are somewhere. You have some understanding. Actually, now we talk about we talk about right shifted curve and left shifted curve. You learn about those things now, right left shifting of the oxygen dissociation curve. Now, when you are getting a curve, so you don't know whether it is a right shifted curve or left shifted curve, right? So because you have only one curve, oxygen dissociation curve. So what you do is now you want to see whether it is a normal in the normal position or whether it is shifted curve, right shifted or left shifted curve. You mark this P50. You draw a horizontal line starting from the 50% and then until it meets the dissociation curve and then draw a line down, right? Vertical line from there. The point that the cross that line in the x-axis is the P50 value. So if that value is 30, so that means your curve is shifted to right. If that is 30, because normally it is 20, 26.7, 26.6, that rate, right? So if it is say 24, so that means the curve is shifted to the left. And for fetal hemoglobin, it is 18, no, because it is grossly shifted to left side fetal hemoglobin. So that is the importance of learning about this P50. Right. So then there are important points. Already we have discussed these important points. I have mentioned when I was discussing. Right. So arterial point, venous point, and the P50. Right? P50. So these are the three important points that you have to mark and you have to talk about. Then you have learned the four types of hypoxia in your medical school as well as during, I think, this postgraduate studies. So there are four types of hypoxic conditions: hypoxic hypoxia, anemic hypoxia, stagnant hypoxia, and histotoxic hypoxia or tissue toxic. This cytotoxic hypoxia, right? So what happened to these various points in where these four types of hypoxic condition? In hypoxic hypoxia, what happens in the arterial blood, arterial partial pressure is low, therefore arterial saturation is low. So therefore, the arterial point is shifted to the left along the curve, right? So that is what happened in the hypoxic hypoxia, right? Because partial pressure is low in the arterial blood because some problem somewhere, right? It could be the lung or it could be the respiratory drive or whatever the problem. So because of that, partial pressure in the blood is in the arterial blood is reduced. So therefore, the point is shifted to the left. But the body try to use the usual amount of oxygen. So there is 250 at rest. So body is trying to extract the usual amount that body wants. So therefore, the venous point moves down. Venous points moves down, right? I think, can you see? Can you see me? Hello? You can see the screen. Can you see me as well? No.
Answer.
Huh? Okay, right. Because when the presenter is not around, you feel sleepy most of the time, no? Right. That is what happened. So because of that, venous point is also moves downwards along the curve. So then in the stagnant hypoxia, actually now stagnant hypoxia means cardiac output is low, right? Like such as in heart failure or even after myocardial infarction. So you might not be producing adequate cardiac output of 5 liters. So you might be producing, say, 3 liters. So if you are producing 3 liters, so that 3 liters circulate in your the vascular system in 1 minute. So therefore, that means the circulation is slow, right? Circulation is slow. So therefore, now oxygen flux is now reduced. Oxygen flux, if you calculate, now earlier you calculated, you have taken the cardiac output as 5 liters, so that is the usual cardiac output. Now say, now here only 3 liters cardiac output. Then what happened? Then that the oxygen flux will be 600 ml instead of 1000 ml, 600 ml of oxygen flux, right? Right. But tissues try to extract the amount of oxygen required to tissues, right? Metabolism. So therefore, venous point moves down, but the arterial point can be normal or low, right? Arterial point can be normal or low in stagnant hypoxia, because arterial point, what happened? So when because if there is no component of the pulmonary congestion or pulmonary edema with this reduced cardiac output, if there's pulmonary congestion or pulmonary edema, then the diffusion will be affected, right? In the lung, diffusion, when the diffusion is affected, so so arterial point will be low. But on the other hand, so blood spend more time in the lung. So therefore, more time is available for diffusion. So depending on which component is more severe, so you will have either normal arterial point or lower arterial point. Right? But whatever it is, only 3 liters cardiac output, therefore oxygen flux is reduced. So therefore, when the tissues extract the usual amount they require within that 1 minute, so venous point moves downward. So that is what happened in stagnant hypoxia. Then in the anemic hypoxia, right? So arterial point is normal, venous point moves down. Again, the oxygen flux is reduced because the partial pressure is normal, cardiac output is normal, but the problem is hemoglobin, not 15 gram percent, but say 7.5 gram percent, right? 7.5 gram percent. So therefore, in anemia, so your oxygen flux is reduced, but you are trying to extract the same amount of oxygen required for tissue metabolism. So therefore, arterial point is normal, venous point is shifted to the shifted downwards. Then in histotoxic hypoxia, the problem is at tissue level, mitochondrial level. So therefore, oxygen consumption is reduced. So tissues cannot utilize oxygen due to cytochrome oxidase problem at the mitochondrial level. So therefore, the venous point is closer to the arterial point, closer to the arterial point, because the tissues are not extracting 250 ml of oxygen. So therefore, arterial point and venous point both are at a closer point, right? Because venous point moves towards the arterial point. That is what happened in histotoxic hypoxia. Those are the what happened during sleep, right? Then you should be able to describe the shape of the oxygen dissociation curve and why that shape is there. So the oxygen dissociation curve has the characteristic sigmoid shape, right? It has the characteristic sigmoid shape. So why it has characteristic sigmoid shape? So because now you should be able to describe why it has this shape and the importance of that shape also you should be able to describe. Now, now, you know that there are this hemoglobin molecule has there are four subunits, right? Each one can bind with one molecule of oxygen. So that means one molecule of hemoglobin can bind with four molecules of oxygen, right? Then combination of the first heme in the hemoglobin molecule with oxygen increases, excuse me. හෙලෝ අනේ සමන් මේ මැඩම්ට කියන්න මම කියලා තියෙන්නේ හොඳේ පැය දෙකක් මම ලෙක්චර් එකක් තියෙනවා හොඳේ මම ඔයාලට මැසේජ් එකක් දාලා ඇති හරි සමන් මේ පැය දෙක කියන්න 11 වෙනකන් හොඳේ හරි sorry because I am on call also but I have handed over 2 hours to my colleague four oxygen molecules can bind. So the combination of the first heme in the hemoglobin molecule with oxygen increases the affinity of the second heme for oxygen. And oxygenation of the second increases the affinity for the third and so on. So that the affinity of oxygen for the fourth oxygen molecule is many times that of the first, right? So when one is bound, so it will increase the affinity, and the second is bound, more and more affinity is increased. So there you have to describe in the molecular basis also. Sometimes it is asked, which will come to come in a few minutes. When most of the hemoglobin molecules are saturated with four molecules of oxygen, no more oxygen can combine with hemoglobin. Hence, the curve becomes flatter. Finally, curve becomes flatter. So that is because no more oxygen can bind with hemoglobin because all the hemoglobin molecules are bound to oxygen, right? So therefore, the curve becomes flatter. The initial part is flatter because the affinity is less. Since because of when the one is bound, the affinity is increases, and the second is bound, affinity is further increased. So therefore, the middle part is steeper, and finally, when the hemoglobin is getting saturated with four molecules of oxygen, then no more can bind. So therefore, the curve become flatter finally. So in the molecular basis also, sometimes some people ask from molecular basis also because quaternary structure of hemoglobin determines its affinity for oxygen. The quaternary structure of hemoglobin determines its affinity for oxygen. So in deoxyhemoglobin, the globin units are tightly bound in a tense configuration. That is called tense configuration. That is tightly bound, which reduces the affinity of the molecule for oxygen, right? Deoxyhemoglobin. So it has a tense configuration. So because of that tense configuration, it has reduced affinity for oxygen. So when the first molecule of oxygen is bound, the bonds held in the globin units are released. So that is producing a relaxed configuration. So exposing more binding sites and tremendously increases the affinity for oxygen. So that is what happened. So tense form is converted to relaxed form when one molecule is bound, one oxygen molecule is bound, it goes to relaxed form. So with that, the affinity for the second, third, and fourth will be increased. So binding on the second oxygen molecule increases the affinity for the third, and therefore affinity for the fourth is increased manifold. So that is what happened in the hemoglobin molecule. The oxygen dissociation curve relates percentage oxygen saturation of hemoglobin to the VO2, right? The characteristic sigmoid shape of the curve is due to the TR interconversion. So that is called TR interconversion. That is called the cooperate binding of oxygen to the hemoglobin, right? So that concept is called the cooperate binding of oxygen to the hemoglobin, right? So you should be able to describe. This is a common fiber question. Right, then a few words about Bow effects. The Bow effect is the decrease in oxygen affinity of hemoglobin when the pH of the blood falls. Right, in the tissues, because of metabolism, so tissue pH is lower, so therefore the affinity of oxygen is reduced. So therefore, it helps in the release of oxygen at tissue level. So, that is about oxygen dissociation curve. Then we will discuss about hemoglobin, right? So hemoglobin sometimes, so we have asked at the Viva, what is hemoglobin? So I am sure all of you know what is hemoglobin, but when it comes to when the question is asked, so you can't answer, right? It is it's a very simple question, but that all of you know. But when the question is asked, so it is difficult to answer. So this is a simple definition for hemoglobin. Hemoglobin is the red oxygen carrying pigment in the red blood cells, right? In the red blood cells, right? So there is a simple definition. So it is a globular molecule made up of 4 subunits, right? Globular molecules made up of 4 subunits. Each subunit contains a heme moiety conjugated to a polypeptide, right, chain. Right? Heme is an iron-containing porphyrin derivative. Heme is an iron-containing porphyrin derivative. So there are 2 pairs of, now there, so in one hemoglobin molecule, so there are 4 polypeptide chains and 4 heme molecule, right? So that means, for each subunit has heme and the polypeptide chain. So there are 2 pairs of polypeptides in each hemoglobin molecule, 2 pairs of, right? 2 pairs of 2 alpha chains with 141 amino acid residues and 2 beta chains with 146 amino acid residues. So that is adult hemoglobin or hemoglobin A. So hemoglobin A has 2 alpha chains with 141 amino acid residues and 2 beta chains with 146 amino acid residues, right? So this is a graphical form. So I have so 4 subunits are there. Each subunit has a heme molecule with iron atom in the center, and then polypeptide chain, right? So altogether, one hemoglobin molecule will have 4 heme molecules and 4 polypeptide chains. In adult hemoglobin, so 2 alpha chains and 2 beta chains. Right. So the functions of the hemoglobin, if you take functions of the hemoglobin, carriage of oxygen, carriage of carbon dioxide, acting as a buffer, and then the buffering of the oxygen in the body, right? So these are the functions of oxygen. Then you can be asked about the other forms of hemoglobin and what are the differences, right? Other forms of hemoglobin, if you take other forms, there are fetal hemoglobin, there's fetal hemoglobin, HbF comprises two alpha chains and two delta chains instead of MRT, right? Then the delta chains have 37 amino acid residues in contrast to 146 46 amino acid residues in beta chains. So right. Then HbS, what happened is HbS? HbS differs from HbA since valine replaces glutamic acid as the sixth amino acid along in the beta chain. It is much less soluble in reduced form than HbA and can lead to abnormal sickle-shaped erythrocytes. So that happened mostly in the venous blood, right? Because right, when the saturation is low, so it becomes sickle-shaped, right? Erythrocyte. So that is what happened in sickle cell anemia. So valine replaces glutamic acid at the sixth amino acid along the beta chain. Then methemoglobin, iron exists in trivalent ferric form. So that is normally it is in ferrous form, and then oxygen can bind with the ferrous form. But here in methemoglobin, it is in the ferric form and unable to carry oxygen, right? Then thalassemia, so it is a hereditary disorder leading to suppression of hemoglobin. So that is adult hemoglobin, suppression of the synthesis of this adult hemoglobin. So therefore, fetal hemoglobin persists to adult life. So that is what happened in thalassemia. So then carboxyhemoglobin, carbon monoxide has 300 times higher affinity for hemoglobin. So therefore, it can easily replace oxygen. So now the carbon monoxide has 300 times higher affinity for hemoglobin. The carbon dioxide has 20 times higher affinity for hemoglobin, right? So now I told you that the amount of oxygen carried by 1 gram of hemoglobin is actually 1.39, but it is in real life, the theoretically actually it is 1.39, but in real life it is 1.34 because now these now methemoglobin can't bind with oxygen, and carboxyhemoglobin also cannot bind with hemoglobin. So therefore, that is why you have lower figure in actual life than theoretical. Right. So one part is over actually now about the oxygen dissociation curve and the hemoglobin. Right. Any question from that part? Common Viva question, nah? Then so carriage of oxygen. So I mentioned about carriage of oxygen, then carriage of carbon dioxide. Now, if you take the production of carbon dioxide, right? Carbon dioxide production in your body. So it is by cellular metabolism. So the amount of carbon dioxide produced depends on the rate of metabolism and the composition of carbohydrate, fat, and protein in your diet. So normally, carbon dioxide, 200 ml of carbon dioxide is produced in 1 minute if your energy is taken from a mixed diet, right? Right. But if you, so oxygen utilized is 250 ml. Now then then the respiratory quotient is 0.8. Now, you know, the normal, so you calculate oxygen, respiratory quotient by dividing carbon dioxide produced by oxygen utilized. So that is the numerator is carbon dioxide produced, denominator is oxygen utilized. So then 200 divided by 250 ml. So the 0.8. So that is respiratory quotient. This respiratory quotient is 1 if you take energy only from carbohydrate. Right? If you are taking energy only from carbohydrate, so it will be 1 because equal amount of carbon dioxide is produced for the usage of oxygen, right? Utilization of oxygen. Equal amount of carbon dioxide is produced because carbohydrate has carbon, hydrogen, and oxygen. When the react with oxygen, it will produce carbon dioxide and water only, right? Carbon dioxide and water. So therefore, equal amount of carbon dioxide is produced. Whereas, so if you are energy is taking only from fat, so then your respiratory quotient would be 7, right? Respiratory, because there are various other elements in fat and protein. So you have various other elements, not only carbon, hydrogen, and oxygen. So therefore, if you have sulfur, it will to burn sulfur. Now you will utilize oxygen, right? You will utilize oxygen, but you will not produce carbon dioxide instead of that. So you will produce sulfur dioxide. If you have nitrogen in the there, so then you will produce nitric oxide, nitrogen dioxide, or nitrous oxide, or whatever the product. So you will utilize oxygen, but you will not produce carbon dioxide instead of that and producing various other things. So that is why when you are taking your energy from a mixed diet, the respiratory quotient is 0.8. So less carbon dioxide is produced than the amount of oxygen utilized. I think that is clear to you. Then if you look at these figures, the rate of transfer of carbon dioxide in the blood. Now in the venous blood, there is 52 ml of carbon dioxide in 100 milliliters of blood. Now in the oxygen, so it is 20 ml of oxygen in the 100 ml of blood and 15 ml of oxygen in venous blood, 100 ml of venous blood. If you take carbon dioxide in the venous blood, 52 milliliters of carbon dioxide in 100 milliliters of blood. In the arterial blood, you have 48 milliliters of carbon dioxide in 100 milliliters of blood. So the difference is 4 milliliters per 100 ml. So the cardiac output is 5 liters. So therefore, each 100 milliliters, 4 milliliters into 5,000 milliliters. Right? So therefore, each minute, 200 ml per minute of carbon dioxide is carried in the blood. Right? So this is at rest. If you take the excretion of carbon dioxide, it plays a greater role in the control of acid-base balance in your body, right? Than your kidney. Because lung is excreting about 10,000 milli-equivalents of carbonic acid. So that as carbon dioxide, you are excreting carbonic acid from your body. So 10,000 milli-equivalents. So I think one book has, says 12,000 milliequivalent. I can't remember whether it is Ganong or Guyton. So 10,000, one book says 10,000, other book says, 12,000, whatever it is a very high amount. So carbonic acid as carbon dioxide per day. But on kidney excrete only 100 milliequivalents of fixed acid per day. So this the equation you can see. So therefore, control of carbon dioxide has a greater control over acid-base balance. But that the renal compensation is more, precise, and the final acid-base balance, it is very important. Then let's see how carbon dioxide is carried in the blood. Carbon dioxide is carried in the blood in three main forms. Dissolved form, so which is responsible for the PCO2 as in oxygen. So dissolved form is responsible for the partial pressure. Then as bicarbonate and also bound to proteins. So three different forms. Now these are the different forms. Right, bicarbonate 90% as bicarbonate. Actually, so this is in the arterial blood. If you take arterial blood, so the 90% of the carbon dioxide exists as bicarbonate. And then in dissolved form, so only 5%. And then as carbamino compounds, only 5%. That is in the arterial blood. Now this bar charts will give you on the left it is arterial blood, right? Arterial blood, like previous diagram, 90% as bicarbonate and only 5% as dissolved form and 5% as carbamino compound. But if you take on the other side, so that is venous arterial difference. This is not the percentages in the venous blood. So it is in the venous arterial difference. Venous arterial difference means that is the amount exhaled into the lung. If you take the exhale volumes from the lung, so 60% has come, right, as bicarbonate. 30% has come as carbamino compound, and dissolved forms only 10%, right? So this is not in the venous blood. I don't know where I couldn't find the venous, values, but it is arterial venous difference. Difference means the exhale volumes into the lung. That is what is given in books. Then we'll talk about carbon dioxide transport. So what happens at the tissue level now under these three headings: transport from tissues to lungs through blood, and then what happens in the lung. So these are the topics that we are going to discuss now. Now here it is a red blood cell, and from tissues to the carbon dioxide produced in the tissues, and that carbon dioxide will diffuse into the red blood cell through the plasma. Then that carbon dioxide inside the red blood cell will react with water to form carbonic acid. That carbonic acid will dissociate into bicarbonate and hydrogen ion. Right? That bicarbonate will be will diffuse into the plasma due to concentration gradient, right? Because concentration of bicarbonate will be increased inside the cell. So hydrogen ion cannot diffuse out, but bicarbonate can diffuse out. So therefore, bicarbonate will diffuse out of the red blood cell into the plasma, right? Due to increased concentration inside So concentration gradient is different. So therefore, to maintain the electrical neutrality between the red cell membrane, so chloride ions will shift into the red blood cell. So that is called the chloride shift. Right? Chloride ions will diffuse into the cells. At the same time, in the tissue level, the oxygenated hemoglobin will release oxygen into the tissues, and then hemoglobin become hemoglobin become reduced hemoglobin, and that reduced hemoglobin can bind with hydrogen ion to form HHB. This HHB react with another molecule of carbon dioxide to form carbon monohemoglobin. So that is the process happening in the red blood cell. Right. So what is Haldane effect? Reduced hemoglobin is less acidic, right? Better proton accepted. At previous slide, I showed you when the hemoglobin is reduced in the tissue level due to the delivery of oxygen into tissues, so it becomes less acidic, and that less acidic means it will accept protons better. So reduced hemoglobin occur when oxygen is unloaded at tissue level. Right? Then hemoglobin become HHB when bound to hydrogen ion. So that HHB bind carbon dioxide at tissue level, and therefore deoxygenation at tissue level increases the ability to carry carbon dioxide. So that is called Haldane effect. So deoxygenation at tissue level increases ability to carbon dioxide. So that is called Haldane effect. So then actually now we have mentioned these three forms of carbon dioxide carriage. So the transport in the blood, so the three different forms, right? So 75% of the oxygen is carried in the RBC, and only 25% of the oxygen is carried in the plasma, right? So this is what we actually discussed: transported amounts and exhaled amount, right? So this is actually exhaled amount. So 60% mostly bicarbonate. Now, this dissolved carbon dioxide obeys the Henry's law that you learn in physics, right? Number of molecules of gas dissolving in the solvent is directly proportional to the partial pressure of the gas at the surface of the liquid. Number of molecules of a gas dissolving in the solvent is directly proportional to the partial pressure of the gas at the surface of the liquid. So that is Henry's law. So the carbon dioxide, dissolving of carbon dioxide will obey this law. So I told you that this carbon dioxide is more soluble, 20% more soluble than oxygen, right? 0.067 ml per deciliter per millimeters of mercury versus 0.003 for oxygen, right? 20 times more soluble than oxygen. So now earlier we saw that 10% of the carbon dioxide evolved into the lungs from the dissolved carbon dioxide, previous one, right? Dissolved from 10%, right? Right. Then as bicarbonate, this is the major form that is carried. So that, yeah, you are familiar with this equation. Carbon dioxide react with water, carbonic acid, and then dissociate into hydrogen and the bicarbonate ion. So this first reaction requires carbonic anhydrase. So carbonic anhydrase, it is found inside the red blood cell. So therefore, the reaction is fast inside the red blood cell. But right. So without carbonic anhydrase, so it is very slow. Right. Do you find carbonic anhydrase in the plasma? Question. If there is an MCQ, right? Carbonic anhydrase is found in the plasma. True or false? True or false? Carbonic anhydrase. I can't see your faces there, therefore you can answer. Right?
True.
No. Only inside the red blood cell, right? So not found in the plasma. So it is mentioned actually so in the respiratory physiology by nuns, right? So not found in the plasma. So therefore, the reaction is very slow in the plasma. So fast reaction in the red blood cell. So the second reaction, that is the dissociation of carbonic acid into hydrogen and bicarbonate, with or without carbonic anhydrase, it is fast, right? So now, so we will come to the carbon dioxide transport in the lung. So the actually exact opposite thing happen in the lung, right? So when you compare with this, what is happening in the tissue level, so the opposite reaction takes place in the lung. So carbon dioxide is lost into the alveoli. The histidine group become more basic. So therefore increases affinity for oxygen. This is one reason for the carbon dioxide, the Bow effect, right? When the carbon dioxide is really lost from the hemoglobin into the alveoli, so it will increase the affinity of oxygen to hemoglobin. So this is what happened in the lung. So the carbon dioxide is lost into the alveoli from the plasma as well as from the red blood cell, right? So at the same time, oxygen is taken up, and then the carbon compounds also will release carbon dioxide and become HHB, and then this HHB will react with oxygen to form like this, right? So exactly opposite reaction will take place in the exactly opposite reaction will take place in the alveoli. So actually, this red blood cells, when it travels in through the pulmonary capillaries, it will shrink slightly, very minute amount. It will shrink because the water also diffuse in the tissues. That when the chloride ions diffuse into the red blood cell, with that it will drag water also into the red blood cell and increases the right size of the red blood cell in the tissue, right? But in the alveoli, when the opposite reaction takes place, chloride ions will shift out of the red blood cell. With that, oxygen, the water also diffuse out of the red blood cell. So therefore, red blood cell will shrink slightly inside the alveolar capillaries, right? So then the bow effect, right? So what happened in the bow effect, right? In the tissues, carbon dioxide diffuses into the blood. So that will increases the partial pressure of carbon dioxide in the blood. So therefore, it will increases bicarbonate and hydrogen ion, and then because of this hydrogen ion, so it will shift the oxygen dissociation curve to the right. So with that means the right shifting of the ODC means the facilitation of the delivery of oxygen. So in the tissue levels, due to this right binding of carbon dioxide, so that will facilitate the delivery of oxygen. So this is what happened. So that is called the Bow effect. That is what happened in Bow effect. Now there sometimes some books describe about this carbon dioxide dissociation curve, but there is no really there is no carbon dioxide dissociation curve, but still some books describe about carbon dioxide dissociation curve. Now, if you look at this carbon dioxide dissociation curve and oxygen dissociation curve in the same graph, so orange color one is the carbon dioxide dissociation curve, and the green one is the oxygen dissociation curve. So if you compare this, so carbon dioxide dissociation curve is situated at a higher level. Then it is more steeper and situated in the higher level and more linear, and so shifted by the oxygen saturation. Now you look at here. So right. So it will be shifted by carbon dioxide dissociation curve will be shifted by the oxygen saturation. So that is the arterial and venous, right? So this is arterial and venous. Right. Any question up to that point? Carriage of carbon dioxide in the blood. Right. Then the regulation of respiration, other part is the regulation of respiration. Now, if you take the respiratory control system in our body, so it has three components. Respiratory control system in the body has three components. There's a central controller, and there are effectors, and there are sensors. So these three components are interconnected or interrelated. Central controller, the effectors, and the sensors. So they are interconnected and interrelated. Right. So the central controllers are situated in the pons medulla and other part of the brain, mainly in the pons and the medulla, but other part of the brain also can control the respiration, right? Mainly in the pons and the medulla. Right. Then this central controller give impulses to the effectors, and effectors give the feedback as well. Right. Effectors. They are the respiratory muscles, mainly diaphragm and the intercostals and other accessory muscles of respiration. So they are the effectors. Then these are sensed by the sensors. So they are the chemoreceptors. And in the lungs and other receptors in various other parts, right? So they give impulses into the central controller, and to control it will give the feedback feedback to the central controller to control respiration. So these are the three components in the respiratory control system. So if you take the central controller, these are the aims of the central control. Why the central control, in other words, functions of the central controller. So the central controller should be able to control involuntary periodical breathing, right? Should be able to breathe while sleeping. Now while you are sleeping also, you will continue to breathe, right? So that is the requirement. So you have to have that function, right? Involuntary periodic breathing. Then you have to have the overriding. That is also involuntary actually. Involuntary overriding. While I am talking, so my respiration is controlled according to the words I am putting out, right? So that is overriding ability. That is again involuntary overriding. So control when eating, drinking, speaking. It will just into the requirement. Right. Then the change output according to demand, right? According to your body demand of your body. So you should be able to control your respiration. So therefore it has to accept information from the sensors. So it accepts information from the sensors and act accordingly. So during exercise and during various metabolic demands like, you have febrile illness or emotion, various. Oh, so this is called malignant hypothermia, right? There is metabolic demand is high. Then additional input from other part of the brain under certain conditions. So like emotional, when you are emotionally, motivated after, especially these young ladies, so they start breathing fast, right? So that is due to, some control from the other part of the brain. So various emotional situations. So they breathe faster. Then central controller, I told you mainly in the pons and the medulla. So it is in the floor of the fourth ventricle. So there are two groups, dorsal respiratory groups and the mainly ventral respiratory group. Then there are pneumotaxic in the upper pons, pneumotaxic center in the upper pons. And then there is a thing called apneustic center. So not very well understood, but so by various research, because you can't cut while you are living and then so study these things. That is the difficulty they have. But from various, analyzing the patients who have got head injuries and all, so they have suggested these things actually. Studying breathing patterns when various part of the brain is damaged by various injuries, right? In trauma patients and all. So they have come to these conclusions. Then the central controller, the two groups of neurons in the pons and the medulla, the medullary respiratory center in the reticular formation beneath the floor of the fourth ventricle. So that is where it is situated, floor of the fourth ventricle. So the dorsal respiratory group is responsible for the inspiration. And the ventral respiratory group is responsible for the inspiration. Right. Then apneustic center in lower pons, causing prolonged inspiratory gasp. So that means, take high tidal volume, right? But the pneumotaxic center control it, which is situated in the upper pons. So it switch off inspiration. So when it switch off inspiration, so it will increases the respiratory rate with reduced tidal volume. right? Then the cerebral cortex can override the function of the brainstem within certain limits. Do you know? Now, if you need, you can voluntarily hyperventilate. You can voluntarily hyperventilate, but there's a limit to that, right? On the other hand, so you can stop your respiration for a short period. Again, there is a limit. After, maximum maybe 2, 3 minutes, you have to restart your breathing, right? So you have a control. So you have control within limits, right? So that is about the central controller, right? Central controller. So mainly in the pons and the medulla, but various other parts of the brain also can influence this control system. Then the effectors are mainly the diaphragm, I told you, and then the intercostal muscles, right? So they are the effectors, abdominal muscles, and accessory muscles of respiration, like sternocleidomastoid and then pectoralis major, minor. So they also sometimes control your respiration in difficult situations. Then, but what is crucially important is where the coordination of these muscle groups, various muscle groups, the well coordination is required uh, between these groups of muscles. Now, you have heard about sudden infant death syndrome or cot deaths. Now, you have heard the word cot deaths. So now it is thought that these cot deaths are due to this, the defect in this coordination because immature brain of the infant, the infant brain is immature. So therefore, that brain cannot well coordinate these functions of these various muscle groups. So therefore, these babies die in the cots. So they can't find any other cause for death. Then the continuing the control of respiration. So under this topic we have covered so far the control of inspiration and expiration, and then now we are going to talk about the control of ventilation for the bodily needs. How the ventilation is controlled according to your demand. So this is central control. So that is for that we have the chemoreceptors. There are central chemoreceptors and there are peripheral chemoreceptors. So in the brain, so we have central chemoreceptors, right? So if you take the central chemoreceptors, so these are the important points on central chemoreceptors. They are located near the ventral surface of the medulla, right? Now your important respiratory centers are central controllers are situated close to that area. So these are also located close to that, right? Near the ventral surface of the medulla. So sensitive to PCO2, but not to PO2 of the blood, right? Sensitive to PO2. Then respond to change in pH of the cerebrospinal fluid, extracellular fluid, so that is same. And when carbon dioxide diffuses out of the cerebral capillaries, right? So these are actually they are sensitive to pH in the blood. But what happened with the carbon dioxide? Because hydrogen and ion cannot diffuse through, but the carbon dioxide can diffuse out of the cerebral capillaries, and therefore it will react with water and then to form finally hydrogen ion. So that hydrogen ion will stimulate respiration, right? So this is how pH and PCO2 affect your ventilation, effect on carbon dioxide and pH on your ventilation. Now, in your x-axis, now you have in the lower line is the pH, the upper line is the PCO2, right? PCO2 in the normal blood, your PCO2 is arterial blood, right? 40 millimeters of mercury in the arterial blood. And the pH in the arterial blood is 7.4. Now you can see the line intersecting all three, that is the normal line intersects with the PCO2 and PO2, right? So in the y-axis, it is the times normal. So 1 is normal, right? So 1 is normal, so it is times normal, right? So alveolar ventilation 1 is normal is the 1 here. So when it is 1, so the partial pressure of carbon dioxide is 40, pH is 7.4, that is normal, right? But you can see from this red line, so there is a partial pressure of carbon dioxide. When the partial pressure of carbon dioxide increase, so there is a massive increase in the alveolar ventilation, right? You can see very steep graph, right? Very, very sensitive to carbon dioxide, right? From so about 30 millimeters of mercury partial pressure up to about 80 millimeters of partial pressure, there's a steep rise in the alveolar ventilation due to rise in carbon dioxide. Right. Then the other green colored line is the how alveolar ventilation increases with the pH. Right. You can see until the pH drops to about 7 millimeters of mercury. So gradually rising alveolar ventilation. But after that point, it started dropping. That is due to direct depression of the respiratory center due to acidosis. When it is pH below is going below 7, so the respiration get depressed due to central depression of respiration due to acid. The same thing happened with carbon dioxide also. When the carbon dioxide partial pressure is very high, the respiratory center will be depressed, and then the respiration become the ventilation become reduced. Right? That is about the central chemoreceptors. Then there are peripheral chemoreceptors. Peripheral chemoreceptors are situated in the carotid. Now there are carotid bodies situated at the bifurcation of the common carotid artery, and then there are aortic bodies that are situated in the aortic over the aortic arch, right? The impulses from the carotid bodies are carried by the glossopharyngeal nerve, and the impulses from the aortic bodies are carried by the vagus nerve, right? Ninth and 10th cranial nerves. So this is how carotid body nerve impulses the rate of carotid body nerve impulses on the y-axis and the arterial PO2 in the x-axis, because the carotid bodies are sensitive to partial pressure of oxygen, right? So when the partial pressure is 100 millimeters of mercury, there is minute, stimulation of the carotid body impulse rate. But when the saturation is reduced until up to 100 millimeters of mercury partial pressure, there is minute increase in the carotid body impulse rate. But when it is below 100, so it starts rising gradually, And then when it is below 60 millimeters of mercury, so that is a critical point. So when it is below 60 millimeters of mercury, the carotid body impulse rate will be increased very steeply, right? Very fast. It will increase the impulse generation, right? So therefore, that is will be responsible for the. Now this response through these peripheral chemoreceptors, that is by due to hypoxia, is very fast, right? Very faster than the central chemoreceptors. But the response is more with the central chemoreceptors, although it is slow. Then this is ventilatory response to carbon dioxide in various situations. Now you can see several lines. On the x-axis, uh, carbon dioxide partial pressure, and on the y-axis, alveolar ventilation in various conditions. The leftmost or topmost line is the metabolic acid. When there is metabolic acidosis, though the response is there's synergistic effect with acidosis. So therefore, the alveolar ventilation is very high when there is metabolic acidosis also. Then the second line is the awake normal. While you are awake, so this is the response you get. And then the other one, while you are sleeping, ventilator response to carbon dioxide is slightly reduced or slightly depressed. The fourth line is the narcotics and the chronic obstruction. That is called chronic obstructive pulmonary disease. So ventilator response to carbon dioxide is reduced. Then deep anesthesia, it is very much depressed. Ventilator response to carbon dioxide is very much depressed. So the lowermost line is the ventilator response to carbon dioxide under deep anesthesia. Now, there are important points that you have to remember in ventilator response to carbon dioxide. So the PaCO2, most important stimulus to ventilation under most condition and is normally tightly controlled. Now, you and I breathe because of your carbon dioxide, because of carbon dioxide in the blood, right? This is a normal condition, so it is the most important stimulants for ventilation. Most of the stimulus comes from the central chemoreceptors, but the peripheral chemoreceptors also contribute to their response too, and their response is faster, right? The response is magnified if the arterial PCO2 is lowered, right? Ventilator response to carbon dioxide is magnified when the arterial PO2 is also lowered. Then ventilator response to hypoxia, important points, only the peripheral chemoreceptors are involved. There is a negligible control during normoxic condition we saw in the previous graph. The control become important at high altitude and in long-term hypoxemia caused by chronic lung disease, right? So this hypoxic drive, when you go to high altitudes, so it will become important because you are hypoxic. And then the chronic lung disease, that is chronic obstructive pulmonary disease, COPD. So you have chronically elevated carbon dioxide, therefore the respiratory center is no more sensitive to carbon dioxide. So your respiratory drive is on hypoxia, right? Then various other reflexes are also responsible for the respiration, the pulmonary stretch receptors, vagus nerve, limit the inspiration. So this is a Erin-Brewer inflection reflex or pulmonary stretch receptors. So this will limit over inflation of the lungs, and there is no adaptation, right? So that will protect your lung, in fact, right? Then there are irritant receptors lie between airway epithelial cells, then stimulated by noxious gases, cigarette smoke, inhaled dust, cold air. The impulses carried by the vagus nerve, and it will cause bronchospasm and hyperapnea sometime. And they are rapidly adaptive. That will mean if you continue to smoke, so these reflexes will be abolished. Then there are J receptors. J means juxta capillary because they are close to capillaries. Juxta means close to. Juxta receptors means they are close to capillaries. Then in the alveolar, the receptors in the alveolar wall close to capillaries. They are endings of the non-myelinated C fibers. So cause shallow breathing in pulmonary edema, right? So in pulmonary edema, you feel some difficulty in breathing due to some congestion. So that is the impulses are carried by these juxtacapillary right, receptors because stretching of these receptors in pulmonary edema. Now you think of how you response in these situations, ventilation is responded in these situations. Diabetic ketoacidosis, what happened? COPD, we have already mentioned what happened in COPD. Then because hyperventilation in DKA, so how it brought about, so you have to think of. And COPD, we have already mentioned. Then acute rise in inspiratory carbon dioxide, what happened, right? So you think of these situations, right? In acute hypoxemia, what are the receptors that are stimulated? And when you are trying to take a tidal volume of 1.5 milliliters, so what happened? Then morphine effect on acute pulmonary edema. So you are blocking some receptors and making you ease, right? Right. So that is about the control of respiration. Any questions up to now? Any questions? Hypocarbia. Hypocarbia is the increase in arterial carbon dioxide levels below 35 millimeters of mercury. Hypocarbia. Below 35 millimeters. What are the causes? The hypoxia is a cause. One cause is because various reasons. If you become hypoxic, so you will hyperventilate, right? You will hyperventilate. We have explained the mechanism in the earlier, right? So because of hyperventilation to maintain oxygenation, right? So you will wash out carbon dioxide causing hypocarbia. Right to left chant, high altitudes and pulmonary pathology. So whatever the cause, so if you are becoming hypoxic, so you hyperventilate and then wash out carbon dioxide causing hypocarbia. Then in metabolic acidosis, right, diabetic ketoacidosis, you hyperventilate because hydrogen ion will stimulate the respiration and then hyperventilate and wash out carbon dioxide. Renal failure, the acidosis is there. Acidosis will stimulate respiration and then hyperventilate. Lactic acidosis, that is due to various reasons when the blood pressure is low, whatever the reason, when lactic acids are produced, tissue hypoxia, and then you will try to hyperventilate. Then various kinds of mechanical abnormalities, sometime in asthma, Actually, early asthma you will hyperventilate, but when the condition is severe and when you are not able to breathe and severe asthma, sometimes you can have hypercarbia also in asthma, but early asthma, so you will wash out carbon dioxide and so hypocarbia, hypocarbia. Then pulmonary edema also, so you will sometimes hyperventilate and then pulmonary fibrosis, right? So you will, so then neurological conditions, head injury, subarachnoid hemorrhage, because of presence of blood in the CSF, the respiration is stimulated and wash out carbon dioxide. Then the effect, what are the effects of hypocarbia? The tetany, one is tetany, so you know what happened in tetany. So due to respiratory alkalosis, so you get this problem and calcium, reduced iodized calcium due to that causing tetany. Then the pulmonary vasodilatation is caused by hypocarbia and thereby reduces or abolishes hypoxic pulmonary vasoconstriction. Then hypercarbia, the excessive carbon dioxide in the blood, they don't give any values, but excess carbon dioxide in the blood or body fluid, hypercarbia. The causes hypoventilation, the COPD we have mentioned before, that is the commonest cause of. Then any cause from the brain stem to the lung, right? So that causes hypoventilation. There are various situations from the brain up to the lung, various muscles, various nerve problems like in Guillain-Barré syndrome. So you will hypoventilate and accumulate carbon dioxide. So up to the lung, various pathologies from the brain up to the lung causing hypoventilation will cause hypercarbia. Right. Then increased carbon dioxide in inspired air. This is mostly iatrogenic. Right? So rebreathing of carbon dioxide if your soda canister is exhausted, or right, then added carbon dioxide in inspired air. Sometimes that also happen, not nowadays, but in the past. Now, you may have seen in some operating theater we have in our hospital the very, very old anesthetic machine with carbon dioxide, the flow meter. So in the past, so carbon dioxide used to recover patients. After anesthesia, they used, now they turned off the anesthetic gases and turn on oxygen and carbon dioxide and to build up carbon dioxide in the blood. So in such situations, so that is why we are telling this rebreathing of carbon dioxide is mostly iatrogenic, right? Mostly iatrogenic. Then increase carbon dioxide production. So you will hyperventilate when you will have hypercarbia. So sepsis leading to pyrexia will cause hypercarbia and malignant hyperthermia. Malignant hyperthermia also produce a lot of oxygen is utilized, a lot of carbon dioxide is produced. And so therefore patient become hypercarbia. Increase dead space, right? Increase dead space, right? So also if the dead space is more than 60%, normally it is 30% of your tidal volume is your dead space. But when it is above 60% of your tidal volume, you cannot compensate. So therefore, you can become hypercarbic. That exceed that. Right. Then the effects on the various systems in the body. Central nervous system, if you take the central nervous system, the cerebral blood flow is increased when there's hypercarbia. Therefore, intracranial pressure is increased because of that. And then the pH changes and affect ventilation. So that will cause an increase ventilation. Then the unconscious, when it is over 90 to 120 millimeters of mercury. Then anesthesia and death when it is above 120 to 150, right? 120 to 150. When it is above 120 to 150 millimeters of mercury, so unconscious or it's like anesthesia and death. Then in the respiratory system, PCO2 above 60 to 75 millimeters of mercury. So patient start hyperventilating. Then the further rise will depress ventilation because due to direct depression of the ventilation due to carbon dioxide. Then in the central nervous system, sorry, cardiovascular system, myocardium, direct depression of the myocardium due to hypercarbia, but there is stimulation secondary to catecholamine release. Because hypercarbia causes release of catecholamine will increase the myocardial contractility and the heart rate, both heart rate and the contractility will be increased by catecholamine. So therefore, indirect stimulation, but direct depression is there in the central nerve. So the net effect will determine the degree of depression or the stimulation, right? Depending on the severity of the problem. So therefore, peripheral vasodilatation will be there with carbon dioxide hypercarbia. Then the tachycardia and tachyarrhythmias due to hypercarbia. Then QT depression and blood pressure on variable effect on the blood pressure because I told you direct depression is there, but indirect stimulation. So therefore, blood pressure will depend on the degree of severity of direct and indirect effect. So therefore, increased pulmonary vascular resistance, I have, written in bold letters because it is very, very important than all the other for you, right? For the anesthetists, increase in pulmonary vascular resistance due to hypercarbia is very, very clinically relevant to you. So that is why I have put, as you know, I have bolded those letters. Because if you, at the exam, if you are writing this, if you somebody asks, these points will get more marks, right? Because more clinically important for the anesthetist will get more marks, right? Hypercarbia and hypocarbia. So I have completed. So then it is straightforward topics, that one. Then fetal oxygenation. Fetal oxygenation. So average fetus is about 3 kilograms. So, but the average fetus will consume at birth. Actually, this is at birth about 3 kilogram, but will consume about 20 ml of oxygen each minute, right? This is also double when compared with the oxygen consumption by an adult. Right? So if you take average adult will consume 250 ml, but weighing about 70 kilograms, 70 kilogram person at rest will consume 250 ml. Then, but this fetus at term is average is about 3 kilos. So, but consume 20 ml. So that is almost double. So if you calculate according to the weight basis. But fetus has to extract oxygen from the maternal blood. Despite this increased consumption, right, double the consumption of the adult, but fetus has to extract that from the oxygen available in the maternal blood. So what are the adaptations for that purpose, right? What are the adaptations? One is the 20-fold increase in the blood supply to the uterus at term. Now, if you compare the uterine blood flow in an unpregnant woman and a pregnant woman, so pregnant woman at term will have 24-fold higher increase in the blood supply. So out of that blood, the major portion is supplied to the placental bed itself. Because uterus is getting about 750 ml of blood at term. So out of maternal cardiac output, 5 liters, about 750 ml goes to the gravid uterus. Right? Out of that, nearly about 50% right, goes to the placental bed itself. So that adaptation. Then high cardiac output in the fetus. Now fetal cardiac output is average weight of the fetus is about 3 kilos, I told you, but the cardiac output is about 1000 ml. So this is very high when compared with 5000 ml cardiac output in adult of 70 kg. 70 kg man has cardiac output is 5 liters at rest, but fetal cardiac output is 1000 milliliters at term. So only 1/5, but only 3 kilos, right? The high cardiac output in the fetus. Hence, the oxygen flux is comparatively very high in the fetus. So therefore, comparatively, oxygen flux is very high in the fetus because cardiac output is very high in the fetus. So out of this cardiac output also, up to 50% goes to the placenta for oxygenation. Now through two umbilical arteries will carry 50% of that cardiac output into the placental bed itself, right? Into the placental bed itself for oxygenation. So then the fetus has hemoglobin percentage of 17 to 18 gram percent, which is about 40% higher when compared with an adult. When you compare with the adult hemoglobin, so that is also an adaptation for survival, the fetal oxygenation. So this also will increase the oxygen flux in the fetus, right? Now you know factors affecting oxygen flux. So the cardiac output, hemoglobin percentage, and the percentage saturation of hemoglobin. Right? Hemoglobin also high, cardiac output is also comparatively high in fetus, so that which will increase the oxygen flux in the fetus. So in the next diagram, actually, I will show you this. As shown below, fetus has HbF, which has a higher affinity for oxygen than adult hemoglobin. So HbF, you know, this HbF has a higher affinity. So that means the P50 is 18 millimeters of mercury in the fetal hemoglobin. So that means higher affinity. So therefore, fetus can extract oxygen from the maternal adult hemoglobin. Then there is a concept called double bow effect operating at in the placenta. So in the placenta, bow effect is operative in both maternal and the fetal circulations. Right? The increase in the PCO2 in the maternal intravenous sinuses assists oxygen unloading. Right? Oxygen unloading from the maternal side. Right? What happened at the placental bed? So carbon dioxide is released from the fetal side. So that will help for unloading of the oxygen from the maternal side. Because when the fetal side, when the carbon dioxide is unloaded, it will facilitate the unloading of oxygen from the maternal side. The decrease in the PCO2 in the fetal side of the circulation assists in the oxygen uploading, right? So when this oxygen, carbon dioxide is unloaded at the placenta, so that will facilitate the uploading of oxygen into the fetal side, fetal hemoglobin. So therefore, the Bow effect facilitate the reciprocal exchange of oxygen and carbon dioxide, right? So double Bow effect means that the ODC for the HbA and fetal hemoglobin moves apart because of this difference in the carbon dioxide, right? So the oxygen dissociation curves will move apart. Now, from here, you can see the adult hemoglobin and the dissociation curve for the fetal hemoglobin and the adult hemoglobin. So therefore, fetal hemoglobin has higher affinity, but with the uploading of the carbon dioxide to maternal side, so maternal ODC will shift to the right side at the placenta, and the fetal hemoglobin, because of the unloading of carbon dioxide, so that will move to the further to the left. So that is called the double Bow effect. These two will move further apart in the placental bed. So that is called the double Bow effect. Right? Is that clear? Double Bow effect? Because there is uploading uploading of carbon dioxide into the maternal side, carbon dioxide into the maternal side, that will shift the oxygen maternal ODC to the right. And then at the same time, due to unloading of carbon dioxide in the fetal side, right? So we'll shift the ODC of the fetal hemoglobin to the further left. So that means that these two curves move further apart, which will facilitate oxygen uploading from the maternal hemoglobin to the fetal hemoglobin. So that is called double Bow effect. So which will help to for fetal oxygenation. Then this is the fetal circulation, the diagram of the fetal circulation. Now you can see the red blood vessels in the red color. So that is the blood coming from the placental bed in the fetal circulation. Placental beds, higher saturation blood comes from the placental bed, but that is not very high. About 80% saturated blood is coming from that side. So which will go to the right, go through the partly that blood will go to the liver, and then the major portion will go to the right side of the right atrium and directly diverted through the foramen ovale to the left atrium, and then that better oxygenated blood will go to the left atrium and then the ventricle, and through the aorta, then it will go to the aorta, and the delivered blood, oxygenated blood into the brain and right upper part of the body. Then what happened? The medium, right? In the purple color. So when it mixes with the blood from the upper part of the body, so it will diverted to the aorta through the ductus arteriosus, and then the saturation in the descending aorta will drop because of that. So that less oxygenated blood will go to the other part of the body. And the venous blood that is even further lower saturation will go and enter into the uh, right side of the heart and go to the right ventricle and deliver to the other part of the body. Whether it goes to the lung, and but that also will be diverted later on to the um, lower part of the body. The coronary arteries originate from the aortic root because in the aortic root, the better oxygenated blood is there. Then the carotid arteries also originate before the ductus arteriosus. So therefore, it will. So those two important organs, heart and the brain, will get better oxygenated blood. And then thereafter, other organs will get the most important metabolic and excretory functions of the fetus are handled by the maternal liver and the kidney. So therefore, the fetus is not dependent on its own liver and the renal function for metabolism and excretion. So that part will be handled by the maternal liver and the kidney. So therefore, the fetus is unaffected by the supply of lower saturated blood to the liver and the kidney. So because heart and the brain will get the better oxygenated blood. So therefore, that is how fetus can survive. So those are the adaptation for the lower saturated blood supplied to the fetus. Any questions? I have come to the end of my presentation. So thank you for listening.
Hello, everyone. Can you hear me and see my screen slides? Can someone unmute and tell me whether you can see? Hello, can you hear me?
Yes, madam.
Yeah, and you can see the screen.
Yes, madam.
Yeah. So everyone is here. I can start. Yeah. 53. Is that the number? Wait for the 2 minutes. Oh, it's started. Okay, good morning, everyone. So welcome to this lecture on isomerism. So I would go through the lecture over about 30 minutes to 1 hour. And during this session, you're free to unmute and ask questions if you don't understand anything at any point. And at the end, I will be